So, hello everyone, I'm Martin Knytl. I'm postdoc from Ben Evans Lab, McMaster University, Canada, and today I will present about sex chromosome turnovers using Xenopus as a model system. So, we have been using Xenopus as a model because this group of animals is highly diverse in the term of sex determination mechanisms. And what are the mechanisms or drivers of sex chromosome turnover? One of these drivers is sexual antagonism. Sexually antagonistic mutations can be beneficial for one sex, but harmful for another. And this is a good example where long tails in peacock are beneficial for males because it attracts females. But long tails would be deleterious for females because it would increase predation. And natural selection favors that some genes are expressed only in males, and sexually antagonistic mutations have the same effect. So sexual antagonism can drive sex chromosome turnover in the following way. Let's imagine chromosome 1 and chromosome 2 in a genome, and there is a sex-determining gene on chromosome 1, depicted by a red color on chromosome 1. And chromosome 1 is transformed to new sex chromosome. And over time, sexually antagonistic mutation can arise somewhere else in the genome. In this example on chromosome 2, and this mutation favors establishment of new sex-determining gene. This sex-determining gene is depicted by red color, and sexual antagonistic mutation is depicted by blue color on chromosome 2. And because sex-determining gene is linked to sexual antagonistic mutation, and this mutation can be favored by natural selection, so chromosome 2 is transformed to a new sex chromosome. And ancestral W chromosome becomes fixed in both sexes because we still need determined femaleness. And several features that are relevant to this model are that sex-specific sex chromosomes becomes autosomes in both sexes, and ancestrally we had female heterogamety, which was transformed to male heterogamety. And this change in heterogametic sex can happen, but it's not required for this model. Recombination suppression also can contribute to sex chromosome turnover. Many of you probably know that natural selection is less efficient when recombination is absent, and this drives sex chromosome, this drives evolution of sex at the first place. Recombination suppression is usually associated with sex-determining locus, and this can cause accumulation of deleterious mutations on sex-specific chromosomes. On this picture, there is a sex-determining gene on W chromosome, and this gene is linked, or deleterious mutations are linked to this, or flattened to this sex-determining gene, and because W chromosome does not recombine with DZ, natural selection is less efficient. And one of the models that are based on recombination suppression is mutational load model. And again, let's imagine chromosome 1 and chromosome 2 on this picture, and there is a new determining gene arises on chromosome 1, which is depicted by red color on chromosome 1. And chromosome 1 undergoes accumulation of deleterious mutations, recombination suppression, and genetic load increases on this chromosome. And chromosome 1 becomes new sex chromosome. But new sex-determining gene can arise on chromosome 2 in this example, and chromosome 2 undergoes, again, accumulation of deleterious mutations, recombination suppression, degeneration, etc. And chromosome 2 is established to be new sex chromosome, and this circle goes on and on again. So one of the features of this model is that ancestral shared sex chromosome Z or X becomes autosome and are segregated in both sexes, and there is no change in heterogametic sex. We assume that at the beginning ancestrally we had dominant female determining gene, which was transformed to another dominant female determining gene. And our research question is how might dominance, recessivity, haplosufficiency, and haploinsufficiency interact to influence sex chromosome turnover. This is a great paper which was published in 1977 by Bull and Charnov, who illustrated many of other drivers of sex chromosome turnover. And one of these drivers or models I am going to describe more in detail. This model is loss of function mutation. So again, there are two chromosomes. On chromosome 1, there is sex-determining gene depicted by red color, and we assume that this gene is dominant and female-specific. But there is another gene which is depicted by blue color on chromosome 2, and this gene is autosomal, present in both alleles in each sex, and we also assume that this gene is recessive and haplosufficient. Haplosufficient means that only one allele of this gene is sufficient enough to influence sex. And over time, loss of function take place on chromosome 2, which is depicted by absence of a blue mark on chromosome 2. And over time, other losses occur on chromosome 2 until only one copy of this gene is present on chromosome 2. But there is still dominant sex-determining gene on chromosome 1. And for the cases this determining gene is not present, which can happen in nature. So we got situation that our haplosufficient allele of recessive gene takes over responsibility for maleness, because these individuals with one allele of this gene develop into males, and if there is no copy of this gene and no dominant sex-determining gene, these individuals develop into females. So one of the features of loss of function mutation model is that this model is independent of sex linkage and or recombination suppression, as both previous models I spoke about are, such as sexual antagonistic model and mutational load model. And change in heterogametic sex can happen. We had ancestrally female heterogamety, which was transformed to male heterogamety. And also we assume that ancestrally we had dominant and female-specific determining gene, which was replaced by the recessive male sex-determining gene. We have been using genome editing, which requires single-guide RNA together with Cas9 protein, and which match target sequence in a genome we want to mutate. And this system introduces double-strand breaks in DNA. And double-strand breaks are repaired by non-homologous enjoining mechanism, and this mechanism introduces deletions in the coding region. And we targeted mutations at the beginning of coding region close to start codon. And our focal gene we have been using is androgen receptor. This gene is important for sexual differentiation. On this picture, it's an androgen receptor protein which recognizes and binds hormone testosterone. And androgen receptor has key roles in sperm maturation, not only in Xenopus but also in humans. And we have been studying tetraploid frog Xenopus laevis, which has only one copy of androgen receptor present in the genome, because it's tetraploid species, second copy of this gene has been lost. And also androgen receptor is autosomal gene, which means that it's not sex-linked in Xenopus laevis. Xenopus laevis females are heterogametic with ZW chromosomes, and males are homogametic with 2 Z chromosomes. So what happens when we knock out androgen receptor in Xenopus laevis? We injected one-cell stage embryos to generate F1 individuals that are heterozygous for knockout mutation, and then we generated F2 individuals homozygous for knockout mutation of androgen receptor, and then we crossed homozygous and heterozygous individuals to generate F3 generation in order to determine sex of this generation. And genetic males that are heterozygous for a knockout mutation develop into phenotypic males, and they were fertile. But genetic males homozygous for a knockout mutation are phenotypic females, and they are fertile. And this means that we reached complete sex reversal using inactivation of both alleles of androgen receptor in Xenopus laevis. And also this means that the wild-type androgen receptor allele behaves like a Y chromosome when it's segregating with a null allele. And this scenario is exactly what Bull and Charnov proposed almost 50 years ago. At the beginning of ancestrally, we had female dominant sex-determining gene, and we know that this gene is the DMW in Xenopus laevis. And because this gene is present only in females, we genotyped sex based on presence and absence DMW gene in Xenopus laevis. And also we had autosomal gene. This gene can be considered as androgen receptor, which was present in both sexes. And using artificial induction of loss of function mutation, we got a situation where only one haplosufficient wild-type androgen receptor allele takes over responsibility for maleness and doesn't require dominant sex-determining gene anymore in order to generate 50% sex ratio. We crossed heterozygous individual with wild-type androgen receptor allele with homozygous individual. The heterozygous individual was males, but homozygous individual was genotypic male, but phenotypic female. So let's say sex-reversed male. And we expect to get sex ratio 50% of male and 50% of females in F3 generation. But these results are still in process. Yeah, because in total, it took me 5 years of animal husbandry and animal crossing of this experiment. And we also have been able to synthesize synthetic sex chromosomes and induce synthetic sex chromosome turnover from female heterogamete to male heterogamete. And more in general, loss of function mutation can lead to change in heterogametic sex, as I showed you, and also loss of function mutation can drive sex chromosome turnover without need of recombination suppression as other models require. And this is another example in nature, which is based on loss of function. So in birds, there is known sex-determining gene, DMRT1, which is present in 2 alleles in males, but only 1 allele or 1 copy in females, because 1 copy of DMRT1 has been lost. And this is not example of haplosufficiency as our androgen receptor allele is, but this is example of haploinsufficiency of DMRT1 allele to determine sex, specifically maleness. And I would like to say thank to my colleagues, which are former students from Evan's lab, Caroline and Shay. And also I would like to thank some collaborators from Marine Biological Lab, where mutant line have been established, Nico and Hitoshi. And thank you for listening. Yeah. I think we have time for one question.
So when you do CRISPR, obviously you can get complete knockouts, but you also might get just a loss of function or a hypomorph allele. Did you see anything like that? And if you saw anything like that, did you get interesting sex phenotypes? Or?
Yes, we did phenotyping. We did histology and phenotype. I did dissection of a lot of organs, such as skull, liver, intestine, stomach, spleen, and I compared this histology of these organs with wild-type females, wild-type males, and heterozygous males and homozygous females. And well, interestingly, we didn't find any differences in phenotypes. Really, like homozygous mutant phenotypic females behaved like normal females, which is very interesting based on mammalian research, for example, in mouses. That, yeah. So this will be also part of the study, but I didn't have time to show you the pictures.
Good afternoon, everyone. I am Sayantan Datta, second year PhD student in the Radcliffe Lab at Georgia Tech. And today I'll be presenting our work on the whole genome duplication in the multicellularity long-term evolution experiment. Whole genome duplications are widespread in nature. We have seen it throughout the tree of life across various domains. Here I'm just showing a tree with the red boxes marking the whole genome duplications in the various domains like plants, fungi, and animals. And they have been shown to be key precursors to rapid species diversification and adaptation events throughout the history of life. And as much as existing polyploid animals are stable, new polyploids that are formed from this whole genome duplications often exhibit genomic instability. We can see from the work of the Gernstein lab here with unicellular yeast that tetraploids very quickly revert back to diploidy in the matter of hundreds of generations. this raises the question of how polyploidy can rise in a diploid population and then be maintained over long-term evolution. Given that the most recent whole genome duplication event in the history of life has still occurred millions of years ago, comparative biology is a bit ineffective in answering this question. So we look towards experimental evolution. And in our lab, we have the multicellularity long-term evolution experiment, where we use the model system, the snowflake yeast. To obtain the snowflake yeast, we take unicellular diploid yeast, delete the ACE2 gene, which prevents the daughter cells from segregating from the mother cells, leading them to form these multicellular clusters that grow fractally in a network. These clusters can grow to a large size, and then after they reach a certain size, programmed cell death or just physical tension can cause them to fracture and form these propagules that can grow as a new individual, leading to the formation of this nice multicellular life cycle, which we then use for our evolution experiment. We have what we call the settling selection regime. We take 10 ml of overnight culture, subsample a portion of it. This subsampling leads to a selection for the growth rate, because if you don't grow, you might not make it to the subsample. Then we let it settle under gravity, and then transfer the bottom 50 microliters for the next passaging cycle. Given that larger things settle faster, this creates a selection for large size, and this is repeated over multiple days. We have 5 replicate populations, which in my graphs you might see as PA125, and it's a long-term evolution experiment, so it's still currently ongoing, and it's in the order of 1,600 days now. And one of the interesting things that we saw from this long-term evolution experiment is that when we first sequenced them, a lot of the SNPs that we identified were present at an allele frequency of 25%. Remember, we started off with diploid ancestors. 25% is not possible in a diploid background. However, it is possible if our system underwent a whole genome duplication during the course of evolution and became tetraploid. To check for this, we had to measure the ploidy of our snowflake yeast. To do so, we first had to generate some controls. So we took our diploid snowflake yeast and genetically engineered tetraploid snowflake yeast. Then we can stain their nucleus with propidium iodide and measure the DNA content using the propidium iodide intensity as a proxy. This will give us 2 peaks for the G1 and G2 cell cycle phases, and where the 4N G1 phase should correspond with the G2 phase of the 2Ns. And using this genetically engineered controls, we can now measure the ploidy of our evolving lines. And what we observe is that not only does tetraploidy arise in our system, it is one of the first innovations to happen during the course of evolution, and it very rapidly, within the matter of 50 days of evolution, takes over the entire diploid populations in almost all of the 5 lines. However, previous work, as I have shown, shows that new polyploids can be genomically unstable. So we checked if this tetraploid is indeed maintained over time, and we noticed that not only does it arrive early, it is maintained for more than 1,000 days and still ongoing, which is about 5,000 generations. So we needed to figure out, why does this happen? What is allowing the tetraploidy to arise, take over our diploid populations, and be maintained? To answer that, we looked at the phenotypes of tetraploids. So we test our genetically engineered system. We see that tetraploidy causes changes in the cell morphology, which is the building blocks of our clusters. Tetraploid yeast forms bigger cells. They also have an increased cell aspect ratio. That is, they form longer cells instead of more spherical cells. Now imagine you're building a thing made out of Lego. If you have bigger blocks, you would build a bigger thing. And if you have, longer blocks and not round blocks, which I don't know if Lego does round blocks, you would actually be able to efficiently use the space available to you to pack more cells, essentially allowing our clusters to not only grow big, but grow to a much larger size before they have to physically fracture. Therefore, we expectedly observe the tetraploid snowflake yeast forming larger clusters. Given that we select for large size, this gives them a huge selective advantage compared to the diploids. So now we know that these fitness benefits can probably help them arise in the diploid populations, but what is helping maintain them in our populations? To check for that, we tried to first remove the selection and instead put a reverse selection. So we grew them on agar pads where we select for small size instead. And we did this to our day 1000 isolates from each of the 5 replicate lines. And we generated more replicates for each line as well. And we observed in almost half of the replicates that we put under reverse selection, tetraploidy is lost. It's either completely lost and they become diploid, or they're partially lost at least. Bringing me to a point where I can answer the first question that we started off with. The immediate fitness benefits of polyploidy can lead to its emergence in a diploid population, and continuous selection on those fitness benefits can lead to its maintenance, despite its intrinsic genomic instability. It's also important to note that the emergence of tetraploids during this evolution experiment is the first observed de novo whole genome duplication in experimental evolution, making the multi a unique long-term evolution experiment of new polyploid organisms, giving me the opportunity to further ask, what is the effect of maintaining polyploidy over long-term evolution? To see what is happening over this 1,000 days or 5,000 generations of evolution, we look at the karyotypes of our lines now. Here, what I'm showing you is a heat map which shows the chromosomal copy number of all the 16 chromosomes of our snowflake yeast. This data is blocked by the various evolutionary time points from which these isolates were obtained. And within each block, we have our 5 replicate lines, which are the rows. What we observe here is that they do show the genomic instability that we expect from polyploids. They become aneuploid. Not only that, at around 600-day mark, there's a lot of convergence in this aneuploidy pattern, especially if you notice chromosome 1, chromosome 8, or chromosome 14, for that matter. The 600-day time point is sort of interesting to us because this is also the time when these lines become really large. They go from forming microscopic clusters that you cannot see with your naked eye in a cloudy solution like that, to forming macroscopic clusters clearly visible to the naked eye. Therefore, I wanted to figure out if this aneuploidy plays any role in the origin of macroscopic multicellularity. It's difficult to genetically engineer such complex karyotypes, so instead we try to do a mutation screen. To do so, I used the unique feature that this macroscopic snowflake yeast have. That is, they form this nice donut, volcano, popcorn. I call them donuts, you can call them whatever, colonies. Whereas we would sometimes see this rare spread out colony, which is unable to form macroscopic clusters. So I took those donut colonies and over a single growth cycle tried to obtain revertants back in that form the spread colonies on plates. The reason for doing it in a single growth cycle is that I did not want any SNPs to occur in the system. A single growth cycle, 5 generations, should be enough for like the chromosomal instability to cause such changes, but given the mutation rate is low, we don't expect any new SNPs. I did this to obtain 25 such replicates from the various 5 lines and 2 different time points, 600 and 1,000 days. And then I tried to compare any karyotypic changes to the karyotypic changes we observed during evolution. So this is the karyotypic changes that we observed during evolution. I'm showing it in a different way. What I'm showing here is, for example, if we focus in the red box, the PA4T600 line, we see that during evolution, the day 600 lines have a gain in chromosome 2. This is during the multi-evolution experiment. Now, I will overlay what we observed during the transition from macroscopic, large size, to small size. If we focus on the same thing again, we see that during the transition from donuts to spread, 2 out of the 3 replicates show a loss in chromosome 2. That is, when we are selecting for large size, they are having a gain in that chromosome. But when we are trying to obtain revertants back to small size, they are having a loss in the chromosome. We see 20 such things across all our replicates, suggesting that certain chromosomal patterns that we observed during evolution were indeed playing a role in evolution of large size. Therefore, we can conclude that tetraploid snowflake yeast become aneuploid during the course of evolution due to the inherent genomic instability in tetraploid yeast. Further, this aneuploid is adding a new level of heritable variation that is facilitating evolution, especially evolution of macroscopic multicellularity. Therefore, on a grander scheme of things, polyploidy can generate structural variations very rapidly, enabling rapid adaptation, which helps us make sense of the events that we see in the tree of life. Whole genome duplication events are an important precursor to, as I said, species diversification and adaptive radiation. Situations where you need rapid adaptation. And what we are seeing is that polyploidy that is resulting from new whole genome duplication events can help in rapid adaptation. There were a lot of people who did this work, so coming to the end of my talk, I would like to thank every one of them, especially Kai, who led the project alongside me, Peter and Ozan, who are the postdocs, Ozan, who does the entire multi for all of this 1,600 days, a test of patience, especially the 4 undergrads who were crucial to this work, and our technician who makes life much easier for all of us. I would like to thank my advisor, Will, and our entire lab, the funding agency, the institute, and all of the work that I presented here today is available as a preprint that you can scan the QR code to see. So thank you, everyone, for listening, and I'll take questions now. Yes.
So you mentioned that the aneuploidy could facilitate rapid adaptation. So do you then potentially see in the multi-lines that the aneuploidy is reversed and replaced by less costly duplications, so that don't require a whole chromosome to be duplicated?
That is an expectation I have that might happen, because a lot of prior work also sees aneuploidy as like a transient response. Though so far, with our 1,000-day isolates, aneuploidy is still existing, and it's existing convergently across lines, so it has not happened. That does not necessarily mean that it won't happen, so probably a year down the line, we might have things where we see less costly mutations arising and the aneuploidy disappearing. But yeah, we don't see it yet. Yes?
Really interesting talk. Is it possible that the gene duplications themselves are the adaptation? So just like packing extra weight into your cells makes you sync faster, and so you don't really find these other phenotypic things?
For tetraploidy, yes, because when you become tetraploid, you have more genome, you need a bigger cell, and so on, which is what we see the cell morphology changing. For aneuploidy, I would argue no, because if you notice in our data, you would see there are gains in chromosome that can cause the macroscopic size to go away, which wouldn't happen if it was just the amount of genome, right? So I think it's the balance of how much of a certain gene or how much of a certain chromosome you have as a whole. Yes?
I have a follow-up question to his question.
Yeah.
First, great talk, thank you. How does, and this may be naive, how does having tetraploidy mechanistically lead to changes and alterations in the form morphology of the cell? I'm not very well versed with that, so I'm curious if you have any hypotheses.
I can't tell you the exact mechanism, but I can give you something that might help understand. Is like, if you have more genome, you'd probably need a larger nucleus to fit all that genome, which in turn you would need a larger cell to fit the nucleus along with all the cell apparatus. You also have more genes, which are probably producing more products, so you'll have a larger cell as a whole. But the exact mechanism of what causes them to become longer, or the exact molecular mechanism of what causes them to be larger, I don't know that, but on a broader scale.
Thank you.
Yes?
Similar to how we didn't have a time at the start, we don't have time at the end.
Okay.
Okay, hi everybody. My name's Colton Siatkowski. I'm a research technician in the Cooper Lab at the University of Pittsburgh, where I recently finished my bachelor's. This talk's probably going to be pretty different from other ones you've seen in this session, but nonetheless, I'm excited to share with you all a newer model for predation of bacteria that we're using in the classroom through our program called Evolving STEM. So before I get into the experiment itself, I just want to give you all a little bit of background about what our program is and what our goals are. So the program was originally made for high school students to simulate the research approach that we use in real research labs. This approach is known to engage students and also increase their content knowledge and their technical skills. And unfortunately, one of the These types of research experiences are often limited to upper level science courses, but one of our main goals is to increase accessibility to these experiences and bring them down to like an introductory biology class level. And since the program's conception, we've also extended down into middle schools as well. All of this is because we are trying to encourage a larger population of students to continue to pursue these science courses and eventually STEM careers, and continue having another diverse generation of scientists to follow us in the future. But in order to do this, we needed an experiment that is both accessible to students that have little to no technical lab experience, but is also reflective of what we do as researchers in the lab. And so we used the Cooper Lab's research on bacterial biofilms and created experiments around that. So for those of you who are unfamiliar, a biofilm is essentially a community of bacteria encased in this structured, self-protected extracellular matrix. And this biofilm concept has been at the center of Evolving STEM's research since the beginning, and an experiment that we still do today with our students that is a classic evolution experiment. Over the course of a week, students will actually see evolution in these populations of Pseudomonas fluorescens. That's a bacteria that we use with these students. And then seeing these biofilm adapted mutants that you can see here. So these mutants rise to a high enough frequency in their population over the course of just 4 or 5 days that they can actually see evolution within their classroom. And I just wanted to highlight 3 mutants here that we commonly see arise from these evolution experiments. So on the left, we have this copycat mutant that actually looks identical to our ancestor, but does produce slightly more extracellular matrix. The fuzzy mutant at the bottom has stronger cell-to-cell attachment to one another and increased biofilm formation. And then finally, on the right, this wrinkly mutant, specifically this WISP-F mutant, produces a lot more matrix than any of these other 3 variants. So taking all of this information together, we wanted to know if these biofilm adaptations could actually protect these bacteria from things like predation, from being eaten from like macrophages, similar to our own bodies. If there's a pathogen, how our macrophages or neutrophils would eat those invasive bacteria. And actually, my PI, Von Cooper, his son, did this as a middle school science project before we started working on it in the lab. I don't know if he got first place. I'm assuming he did. So this new experiment that we're doing, the predation experiment using the slime mold Dictyostelium discoideum, or Dicty for short, as a model predator, since it also consumes bacteria using phagocytosis, just like macrophages in our own bodies. So Dicty is a very unique specimen in that it is normally unicellular, but forms these complex social structures under starvation. So this video that I'll start on the right was taken by another undergraduate, his name's Sachin, that I worked closely with this past year. So we'll kind of go through what's happening in the video along with the social cycle on the left side of the screen. So this is a lawn of bacteria with Dicty on it, and they are forming these mounds and motile slugs that you can see moving around. They form these social structures when they have run out of food. So they're going to start forming these fruiting bodies that you'll see in like the bottom center of the screen. Those stalks are starting to rise up, and then there will be spores that form within those that will be dispersed out so that a new generation can be propagated and more bacteria can be eaten. And this video was time-lapse. It was over the course of about 2 days, I think. Sped up, so it's like 40 seconds. So how we actually conduct this experiment in the lab and in the classroom, we make a lawn of bacteria on these media plates and then put Dicty in the center, just using these filter discs. And over the course of several days or a week, every day we'll outline the area that Dicty has eaten the bacteria just using a marker. And then at the end of this period, however long it takes for them to eat the bacteria or however long we're able to be in the classroom, we can then analyze the clearing rates of Dicty on a specific variant and then this is also a time when we're encouraging students to communicate with one another and share their results as we do in a research lab, because this is something that they don't necessarily have a lot of experience doing, especially in middle schools where we're doing this experiment a lot. So these are some results. I apologize, I don't know if you can see on some of the graphs, I plotted the ancestor against 3 of the mutants that I showed you earlier. The copycat is eaten slightly faster than the ancestor, and the fuzzy slightly slower. But what I really hope you get out of this is that the wrinkly mutant is eaten much slower than any of the other 3. And so from this, we were wondering if it was specific to this WISP-F mutant that these biofilm adaptations would be protecting it from predation. And so we have another wrinkly mutant mutation in the WISP-A gene. And we actually gave these WISP-A mutants to students to use to kind of get preliminary data from in a school. And they found that the mutants were eaten at a similar rate or slightly faster than the ancestor. And of course, we had to repeat this in the lab because things are not sterile at all in public schools. But we did find actually the same thing, that the mutant was eaten about as fast, if not faster, than the ancestor, and certainly faster than the WISP-F mutant. But this is not totally surprising to us. As you can see in this little chart at the top, we have relative biofilm production as something that we're able to measure in the lab, and the WISP-A mutant does not make as much biofilm as the WISP-F mutant, but this is something that we're still investigating. So while students were excited that they got to work with this new mutant, one of the other things that we like to gauge how they feel about their experience with science in general, especially science labs, since that's really what we're there researching. So we have some statistics that we've been able to gather from this past year. So we work with a very diverse group of students. These are all from Pittsburgh public schools that we worked with. And this is just the dumbbell plot showing student responses to pre and post surveys before and after doing this experiment. They respond on a Likert scale of 1 to 4. So 1 would be strongly disagree and 4 would be strongly agree. So I just want to draw your attention to a few of these specific questions. There is a significant increase in all student demographics agreeing with the statement, "I am a scientist," after doing the experiment, which I think is exactly what we want to be seeing. We're promoting, the love of science and hoping that they're getting to see themselves as scientists, which we see here. Next, they have an increase in some demographics agreeing with the statement, "I often ask questions about science," which I think is somewhat indicative of how much this experience is allowing these students to reflect more on themselves and their own experiences, using more critical thinking skills and wondering what's actually going to happen rather than just reading about an experiment in the textbook. One more slightly disheartening one that we see up here. They have declined a little bit, saying that they don't want to find out more about science, which is not what we want to hear. But it's still good for us to know, because that just means that for some students, this was not the most conducive way for them to learn. And we do verbal post-interviews after the experiment where they can tell us how they felt, what they think we could improve upon, and we're always trying to do better things for these students as well. So similarly, these are student views of science labs rather than how they feel about science itself. And just one to highlight on here, after doing the experiment, they would say, "I know more than my friends about doing science labs." In some cases, they feel that their friends are not so smart after working with them. But I think particularly with female identifying students, there is a significant increase, and I think this is really important in understanding that perhaps they were not feeling so confident in themselves being in the STEM fields, but after doing this experiment, they are able to see themselves going forward, taking more advanced science classes and doing a STEM or STEM-related career, which is another core thing that we're trying to promote. So this map shows all of the schools that we've been in. We primarily work in the Pittsburgh public schools, but we have schools that have done this program all over the country, both high schools and colleges or universities, and then a few middle schools as well. But what's next for us, we are actually, every summer we do teacher training kind of seminars every day. So we work with the teachers, teach them how to do these experiments, and then go into the classrooms with them in the following school year. So we'll have more teachers to work with. That obviously gives us more students to work with, and we'll work with the same students that we did in the past years as well, and more biofilm adapted mutants. As I showed you earlier, we were able to get that preliminary data from that one class of the WISP-A mutant, and students, we never know what they're going to find, so we're looking forward to that. So I just want to thank my mentor, Dr. Abby Matella, up here for everything she's done for me the past year and encouraging me to come and give this talk. Vaughn Cooper for allowing me to come here and do this presentation. Sachin, Sarah, and Sophia, they contributed a lot to Evolving STEM or the data that I showed you here today. And our colleagues in the University of Pittsburgh School of Education, and of course all of the students and teachers that allow us to do this research. Thank you. I'll take questions. Yes.
Great talk. I was just curious about having students identify more as scientists and them clearly showing like an interest in science, but simultaneously not wanting to learn more.
Yes.
Like, what's the plan to try and address that?
Yeah, that's a great question. Thank you. I think, as I showed, there are very diverse groups of students in every school that we work with. Even though it's within the same school district, every school is diverse in a different way. So we're doing these interviews with these students and seeing, how do they personally feel about how they're viewing themselves and the experiment. And in some cases, it's the environment that they're in addition to the program itself. So there's a lot of different variables that we're working with and trying to incorporate into our analysis. Yeah.
Well, that was awesome.
Thank
Great
you.
job. This looks really cool. I have kind of a silly question. Do you know which is the school district that's in Northwest Ohio?
I do not personally.
Yeah, I'm sure you're not going to all of them. But I'm from that area, and it's a really, really rural area. And all the schools are really poor. And I went to those schools, and I can say that there is nothing even close to this. In my school, I graduated in 2007. We were reading science books from the 80s, and there were no experiments, ever. So this is awesome. Great job.
Thank you.